Thank you very much once again. Let me welcome you back to this one channel. And then today is a continuity of now grade 12 life sciences. And then we are saying we are under term 4. And then this is a revision for protein synthesis. Ensure that you are hitting the subscription button and ensure that you are sharing to those who are concerned patient into this protein synthesis. Remember that in grade 10, you've made protein synthesis. And then here in grade 10, remember that we have made the structure, one of the key concept or the key adaptation of the cell. The function of the cell, it is to promote life, and the organs, they are the nucleus, which can be found on the DNA. Remember that number one, we have said that we are having on the cell, the nucleus of the cell consists of the nuclear membrane, the nucleoplasm, the nucleolus, and the chromatid network, which forms later on into the chromosomes. This is one of the key concepts that we have learned on our previous grades to say we have now these labelings, and they play a vital role. And then number two, we have talked about the structure of the RNA and DNA, and then where is it found in RNA? And then it is found on the nucleus and the cytoplasm of the cell, and it plays the role in the protein synthesis. So we are talking about the protein synthesis, of which later will describe the translation and the transcription process. And then number three, we have talked about now the DNA, we say, and it forms part of the chromosomes. So we are saying that these are the genetic carriers. These are your genes. How are you going to look like phenotypically and genotypically? Remember that you are carrying 23 genes from the chromosome from the paternal side and the maternal side, which can lead to 46 chromosomes. And then let us talk about number three, the ribosome. We are saying that for protein synthesis to occur or to facilitate better, it needs the ribosome and are mainly found in the cytoplasm of the cell and are the sites of the protein synthesis. When we are talking about the site, we are saying the location where the protein synthesis occur and happens. If ever we are moving on again, we are seeing that we can be asked to define the concept of protein synthesis under section A. And then we are saying it is the process whereby proteins are made in living cells of the organisms to form enzymes. Remember that we have said that enzymes, these are chemical reactions which fastens the process. And then they triggers hormones, and these are chemical reactions which they make the facilitation of the proteins to be optimal and to work effectively into the cell. Because of the cell, it carries life. We are saying that amino acids are the basic building blocks of the proteins, which they are the monomers. And then the nucleic acid, the DNA, and the RNA are involved in the protein synthesis. The main step of the protein synthesis are the transcriptions and the translation. The transcriptions and the translation are the major two concepts which will later learn into this structure and how they can be asked in the examination. We are assuming we can see that we are having now the transcription on the transcription side. So we are saying this is the DNA, and the DNA, it is found where? In the nucleus. So we are saying the nucleus, it carries the DNA. How are you going to look like? And then we have DNA, it will unzip just a little bit and unwind, causing the mRNA in the codons into the triplets to merge or to be found in these codons. And then we are saying under this, it is found where? In the cytoplasm. Before we can go on, we can see that when this unwinds, we are saying the template, it is going to be carried away, and the codons and anticodons, they are going to match each other. And then remember that this order, we are saying that order DNA is the adenine, the guanine, and the thymine. The RNA codons is the uracil, synthosine, and adenine. The tRNA, so it is found where? In the cytoplasm. So we can see that under this process, what is going to happen is going to occur under the transcription, whereby these codons, they are going to match, or they are going to match with the other that they can be matched with. So the uracil, it will go with the adenine, the xanthosine, it goes with guanine, and the adenine, it is going with the uracil. And we are saying it happens where? In the ribosome of the person. And then we are saying that under this, we can understand that the translation process will therefore match with the tRNA and the anticodons that are available, causing this protein synthesis to happen. All right, to see that we are having questions based on this concept that we have been teaching up until to now. Under this, you must understand that one of the typical questions that can arise there, it is to name and describe the process occurring in the nucleus in which it results in the formation of the mRNA. Understand that the process, it is the transcription, and the transcription, it happens, number one, the process is transcription, you get the mark. Number two, the double helix DNA molecule unwinds. Number three, when the hydrogen bonds break. Number four, the DNA molecule, they will unzip. Second DNA strand, they will be separate, which I've shown you on the picture, the diagram. Number five, one strand is used as a template to feed for the mRNA using the free mRNA nucleotides from the nucleoplasm. The mRNA will go synthesis, and then this process is controlled by the enzymes, which they fasten the process to happen faster. All right. And then in number two, it says now describe the process of translation. How does it occur? We are saying each tRNA carries a specific amino acid, and tRNA carries amino acids to the ribosome. When the anticodon on the tRNA matches the codon on the mRNA, the amino acid becomes attached in the sequence determined by the mRNA by the peptide bonds to form the required protein, so that it is for 5 marks. All right. And then those are the typical questions that they can teach. And then if ever we are moving on again, we are to see that we can have some other questions which they are correlated to the concept. In this instant, we are going to see this question. And let us read now the statement that is given. A species of bacteria contains a type of protein called protein 1. A mutation. What is a mutation? This is a genetic disorder that can happen. It can occur which resulted in the formation of the second type of protein called protein 2 instead of protein 1. The scientists determined the amino acid sequence of each protein. They then used the amino acid sequence to find the DNA base sequence that coded for the portions of this protein. The results are shown in the tables below. So we've got the portion of, and then we've got the portion of protein 2. Under this, we must understand we've got the amino acid sequence and the DNA base sequence. And then we are saying the lysine, it must be the TTT, which now would represent this particular DNA base. And then the TCA, which is a thymine, sericyle and adenine, cytosine and adenine. This is guanine and thymine, adenine, cytosine and guanine. So in the portion 2, the sequence would also be the same, ensuring that these amino acid, they are in the same portion of the proteins. One of the key concepts of the questions that we can be asked in the examination, it is to say, give the DNA triplet for the third amino acid from the left in the sequence for protein 2. So in the left for protein 2, this is the one in protein 2 from the left. And then we are saying it is the answer. It is guanine, and thymine. And then if ever we are moving on again, we can see that give the codon for the lysine. And lysine, it is adenine, because of the lysine is here. So we are saying this thymine, the answer there, it will be adenine, that they would be merging. And then if ever we are moving on again, we can see that the question, it says, give the anticodon serine. And then the anticodon serine, it will be uracil, syntacine, and adenine. And where can we find that? This is a synthesine. And then if the serine is here, and then that is where we can be finding these answers that they can match. And then if ever we are moving on again, we can see that the here, this uracil, it will go with the thymine, again with the guanine. And number 4, it says describe how the mutation caused by a change in the structural protein. All right. And then if ever we are moving on again, we are to see that number 1, one base triplets to the strands has changed from ACG, which is the adenine, synthosine, and the guanine, to adenine, synthosine, and synthosine. The triplets ACG codes for the amino acid synthosine. The triplet ACC9 synthosine, it codes for the amino acid tryptophan, and then this result in the change of sequence of the amino. You have heard the lesson usefully, and then you can ask a relevant question on our comment section. Thank you very much for your time.